What if we could genuinely blur the lines between life and machines? I hope you enjoy our video.
Okay, here's a thought to start us off. What if we could genuinely blur the lines, between living things and machines? Like, what if AI wasn't just designing software, but actually designing life itself? Today, we're doing a deep dive into Xenobots. They're this fascinating new class of artifact that really challenges everything we thought we knew about robots, about living systems. We've gathered a whole stack of sources to really unpack what they are, how they're made, what they can do, which is pretty incredible, and also the big implications, both the good and, well, the challenging. So our mission today, give you a clear, concise shortcut to understanding this really revolutionary science.
Yeah, and it's crucial to get this right from the start. We're not talking about traditional robots here, metal, plastic, wires, none of that. These are actual living, programmable organisms designed by AI, built entirely from biological cells. They get their name, Xenobots, from the African clawed frog Xenopus laevis. It's the embryonic cells from that frog that are the basic building blocks.
Wow, okay. Living, programmable organisms. That's a fundamentally different concept. So let's dig into that. What exactly are they made of? How do they work at that core level?
Right. So at their essence, Xenobots are synthetic life forms. Computer design, like we said, then hand assembled, but every single part is living biological tissue. The sources are really clear on this. They are neither a traditional robot nor a known species of animal, something entirely new.
And it's a common mistake to think they're genetically engineered, right?
Yeah.
People hear synthetic life and jump to that conclusion, but they're just regular frog cells rearranged.
Exactly. That's a key point. The DNA is 100% standard frog DNA. Xenopus laevis, no gene editing involved. The real breakthrough, the novelty, comes from the physical organization, the morphology. It's about arranging those standard cells in radical new ways. Think of it like having the same Lego bricks, but building something completely unexpected, something the original instructions never envisioned. It's about tapping into what the researchers call the morphogenetic code, treating the physical shape itself as something you can program.
So if you can program biology just by rearranging things, does that mean life isn't just about the genes? Is there like an underlying logic of physical form we're just starting to see?
That's precisely the implication, and it's huge. It suggests there's this whole other layer of information in biological systems beyond just the genome. It's about how cells cooperate, how they collectively build structures. It's like discovering the operating system that runs the genetic software.
That is mind-bending, the
Yeah.
potential there. Like debugging developmental diseases by correcting the assembly instructions.
Potentially, yes. Or building complex tissues and organs from the ground up by understanding those assembly rules. Xenobots really force us to rethink biology at a fundamental level.
Okay, so how do they actually make these things? You mentioned AI design and hand assembly. Tell us about this design-build pipeline. It starts in silico, right, with the AI.
That's right. Stage one is the digital design. They use an artificial intelligence specifically, an evolutionary algorithm running on a really powerful supercomputer, like Deep Green. This AI designs literally thousands of virtual body shapes. It's testing them for specific tasks, like moving effectively or pushing tiny particles around. It runs through billions of configurations over months. And the designs it comes up with are often, as the sources say, very non-intuitive, shapes a human would probably never think of, but they turn out to be super efficient.
So the AI spits out these weird, efficient blueprints. Then the biologists take over for the in vitro construction. How fiddly is that?
Oh, it's incredibly intricate. Think microsurgery, but even smaller. Biologists harvest those early stem cells from frog embryos. Then, using tiny forceps and electrodes, they meticulously sculpt the AI's design cell by cell. They place passive skin cells to form the structure, the scaffolding, you could say. And then they strategically position contracting heart muscle cells. Those provide the rhythmic pulses for movement. It's painstaking work, building these millimeter-scale living machines.
Millimeter scale.
Yeah.
And once they're built, they're just
Yeah.
autonomous. They run on their own.
Yeah, completely. They're powered by internal energy reserves, lipids, and proteins stored within the cells, enough to keep them going for about a week. And this is a huge, plus they just biodegrade. They turn back into dead skin cells. No pollution and no electronic waste.
Okay, that's already impressive. Biodegradable, autonomous,
Right.
like the self-repair thing. That sounds like science fiction.
It does, but it's real. If you cut a xenobot, the cells around the wound spontaneously work together to stitch it closed. Within minutes, they restore the original shape and, crucially, the original function. It's not just patching a hole, it's true biological regeneration, a property metal robots just don't have.
That self-repair is
Yeah.
just, yeah, Astonishing. And the pace of this research has been really fast, too, hasn't it? From the first ones in 2020.
Absolutely phenomenal speed. Xenobot 1.0 back in 2020 was really the proof of concept. Manually layered heart and skin cells. They showed they could move, push little pellets, even sort of cluster together and push debris into piles, emergent swarm behavior. Lasted about a week, self-healed, like you said.
Then Xenobot 2.0 came along in 2021. That sounded like a pretty big step up. What changed?
It was a significant leap. They moved from manual layering to more of a bottom-up self-assembly. They use epidermal progenitor cells, basically, very early skin cells. And these cells spontaneously grew cilia.
Cilia, like tiny hairs.
Exactly, tiny hair-like structures that beat, providing much faster, more agile movement. It was about letting the cells use their own innate abilities. It really highlighted this concept of cellular plasticity, how cells can adapt and take on new roles based on their environment and neighbors.
And 2.0 also introduced a kind of memory, didn't it, that hinted at more complex stuff down the line.
Yes, that was a fascinating addition. They engineered the cells with a fluorescent protein called EOS-FP. Normally it glows green, but if you hit it with a specific wavelength of blue light, it permanently switches to glowing red. So if a xenobot swam through a patch of blue light, it would turn red and stay red. It basically recorded that event, a simple one-bit memory. It was a really crucial proof of principle, showing you could build biological sensors into these things, a first step towards information processing in a living machine.
Okay, sensors, memory. That's already complex. But the real headline grabber, the one that got everyone talking, was Xenobot 3.0 later in 2021. The kinematic self-replication. That just sounds revolutionary.
It fundamentally is. It expands our whole definition of what reproduction can even mean. We're used to biological reproduction happening internally, right? Cell division, budding, sexual reproduction. It all starts inside the parent. Kinematic self-replication is totally different. It's based on external movement and physical assembly. The parent xenobots act like tiny construction workers. They actively gather loose cells from their environment and compress them together to build functional copies, the offspring. It's reproduction by building, not by dividing. Unprecedented at this scale.
Reproduction by building.
Right.
Okay, how does that actually work? I remember reading the first attempts with the simple spherical ones weren't very good at it.
That's right. The initial spherical xenobots, developed from the 2.0 platform, would sort of randomly bump into loose cells and push them into piles as they swam around. Sometimes these piles would stick together and form smaller, functional offspring, but it was really inefficient. The offspring were often too small or poorly formed. The whole process usually just fizzled out after one, maybe two generations.
So the AI had to step in again to solve this replication puzzle.
Absolutely. They tasked the supercomputer again, this time optimizing the body shape specifically for efficient replication. Months of simulations, testing countless forms, and the shape it converged on was, again, very non-intuitive. It was a C-shape, like a little semi-torus, and everyone immediately saw the resemblance.
Pac Man.
Exactly. It looked uncannily like Pac Man. It's kind of amazing that an AI with zero cultural context would arrive at that specific, highly functional shape.
So these Pac Man Xenobots were much better builders.
Dramatically better. That C-shape, that concave mouth, was perfectly suited for the job. They actively used it to efficiently sweep up, corral, collect, and then compress the loose stem cells into dense spherical offspring. This optimized shape allowed the replication process to continue for multiple generations, up to 4 or 5, before eventually stopping. A huge improvement in efficiency.
Okay, but this whole replication thing did spark some debate, right? Some critics felt it was maybe being oversold.
Yes, there was definitely some pushback. The main arguments were, 1, it's entirely dependent on that external supply of loose cells, the feedstock. It can't replicate without those raw materials being provided. And 2, the offspring aren't exact copies. The C-shaped parent makes spherical offspring, so it's not cloning in the way we usually think of it.
Right, not like bacteria just dividing into identical copies. But the researchers pushed back on that critique.
They did. While acknowledging those points, they strongly argued that it's still a novel and significant form of replication, completely unknown before at the cellular or organismal level. Their key point is that it reveals this hidden potential within cells. These frog cells have a genetic program to become a tadpole, but freed from that developmental path, placed in this new context, they tap into something deeper, maybe more ancient. They exhibit this collective intelligence, this plasticity, to solve a problem replication using physical means, using their morphology. As one researcher put it, these cells have the genome of a frog, But freed from becoming tadpoles, they use their collective intelligence to do something astounding. They're building new life from spare parts.
That's a powerful perspective. Okay, so with all this astounding science happening, what does it mean for the real world? Where could this actually go?
Well, almost immediately after the first Xenobot papers came out, this really compelling vision took hold in the public imagination. The idea was deploy swarms of tiny biodegradable Xenobots into the oceans to find and collect microplastics, corral the pollution. And because they're biodegradable, they wouldn't add to the pollution problem themselves. It's a beautiful idea, very sci-fi.
It sounds fantastic. A biological cleanup crew for our plastic mess. But how realistic is that? Is it something we could actually do, say, in the next decade?
Honestly, it's a powerful aspirational goal, but as a near-term solution, it's just not feasible. There are some major fundamental hurdles. First big one, the saltwater barrier. Xenobots are made from freshwater frog cells, put them in salt water, and the osmotic pressure difference basically kills them very quickly. They just can't survive. Maybe future versions could use saltwater organisms, but that's a whole different research challenge.
Okay, so the environment is wrong.
Right.
And even if it wasn't, the scale. The ocean is vast.
Exactly. That's the scalability chasm. Cleaning microplastics from the ocean would require trillions upon trillions of xenobots, maybe more. Right now, making them involves painstaking microsurgery or very controlled lab-based self-assembly. We have absolutely no way to mass produce them on an industrial, let alone planetary scale.
And wait, they don't actually eat the plastic, do they? They just hurt it.
That's a functionality gap.
Yeah.
Yes, they can push things around, herd particles, but they don't degrade or consume the plastic. So even if they could collect it all, you'd still need a completely separate system to actually remove those piles of plastic from the ocean. Another massive challenge.
Okay, hurdles piling up. What else?
Energy and lifespan. They run on stored energy for about a week. The open ocean is mostly a nutrient desert. There's no way for them to refuel or sustain long missions out there. And finally, control and retrieval. How would you even track, command, or collect microscopic, biodegradable robots spread across millions of cubic kilometers of ocean? We simply don't have the technology.
Right. So the ocean cleanup dream, while inspiring, is firmly in the realm of science fiction for now. It's important to separate that hype from the reality. So if not the oceans, where is the real near-term potential? Where could xenobots or similar biobots actually make a difference soon?
The much more realistic and frankly exciting near-term potential lies in contained, controlled, high-value environments. And the absolute front-runner is biomedicine. Think about it. The human body is a much more suitable environment. It's aqueous, temperature-controlled, nutrient-rich, much closer to the conditions they need.
That makes total sense. So what specific medical uses are we talking about?
Several really promising avenues. Targeted drug delivery is a big one. Imagine programming tiny biobots to navigate your bloodstream directly to a tumor or the site of an infection and release a drug payload right there. This could drastically increase treatment effectiveness and reduce the awful side effects you get when drugs flood the whole system.
Like tiny guided missiles for medicine. What about the idea of them acting like miniature surgeons?
Exactly. Their small size and motility could allow for internal surgery at a scale we can't currently reach. Maybe scraping plaque from the inside of arteries without invasive procedures, or clearing blockages in tiny capillaries in the brain or eye, performing tasks too delicate for human hands or current instruments.
And you mentioned building them from a patient's own cells. That seems crucial for avoiding immune rejection.
Absolutely essential. This leads directly into regenerative medicine and the development of what are being called Anthrobots, biobots made from human cells. If you build them from a patient's own cells, like tracheal cells, which they already experimented with, the body shouldn't reject them. These patient-specific bots could potentially guide tissue regeneration, help heal injuries, maybe even assist in building engineered organs. And there was this really interesting finding where Anthrobots seemed to encourage damaged neurons to heal in lab tests, hinting at possibilities we haven't even fully explored yet.
Wow. Beyond direct treatment, any other roles?
Yes, they're also potentially powerful tools for disease modeling and drug discovery. Instead of testing drugs on flat 2D cell cultures, you could use these dynamic 3D living systems to better simulate how cells interact in tissues. This could lead to more accurate predictions of drug effectiveness and toxicity, speeding up the drug development pipeline.
Okay, so biomedicine looks like the major focus. Any potential for that environmental cleanup idea? Just maybe, a smaller scale.
Yes, potentially in contained environmental remediation, not the open ocean, but controlled environments. Think wastewater treatment plants. Xenobots could be designed to target specific pollutants, heavy metals, or harmful microbes, clumping them together for easier filtration. Or maybe cleaning up toxic spills in contained sites, identifying and consolidating hazardous materials safely. The keywords are contained and controlled.
Yeah.
And, connecting back this pivot to using human cells for Anthrobots while medically promising, it really dials up the ethical questions. We start talking about identity, consent, ownership. It moves from research tool to something deeply personal.
That's a perfect transition. Because whenever this kind of tech comes up, the first fear people often voice is, well, the sci-fi nightmare. Runaway replication, gray goo, self-replicating biobots taking over.
It's an understandable fear fueled by fiction, but based on the current Xenobot 3.0 technology, those scenarios are highly improbable. There are several built-in safety factors. First, their environmental fragility. They need very specific, sterile lab conditions. They wouldn't last long out in the wild. Second, the feedstock dependence we talked about. Replication grinds to a halt immediately without that constant external supply of prepared stem cells. Third, their finite lifespan. They naturally biodegrade in about a week. No long-term persistence. And fourth, all the research is contained by design. It happens in secure labs, under strict protocols, reviewed by ethics committees.
Okay, so the attack of the Xenobot scenario isn't realistic right now. Phew. But that doesn't mean there aren't real ethical issues we need to grapple with, does it? What are the more grounded concerns?
Absolutely. The real ethical landscape is complex. One major area is dual-use potential. The same capabilities that allow targeted drug delivery could, theoretically, be repurposed to deliver toxins or pathogens. That raises concerns about bioterrorism or biowarfare, especially if the tech becomes easier to access the garage biobot idea. Then there's the deep philosophical question of ontological and moral status. As these biobots potentially become more complex, maybe incorporating nerve cells down the line, questions about their status arise. Do they experience anything? What duties do we have towards them regarding their creation, use, disposal? What do we owe these novel entities? And finally, there's governance and regulation. This technology sits right at the intersection of AI, robotics, and synthetic biology. It doesn't fit neatly into existing regulatory boxes. There's a really urgent need to develop new, flexible international governance frameworks that can adapt as the technology evolves, involving scientists, ethicists, policymakers, and the public.
That's a huge amount to think about. Governance, moral status, dual use. What stands out to you as maybe the biggest, most immediate ethical challenge we need to address? Something for all of us listening to really chew on. All right, so let's try and pull the threads together from this deep dive. Xenobots are, without a doubt, a monumental scientific leap. They're changing how we even think about biology and engineering. But that popular vision of them cleaning the oceans, inspiring, yes, but for now, it's more science fiction than near-term reality.
Right. The true revolution, the real value right now, is Xenobots as this unprecedented scientific platform, a tool for exploring the morphogenetic code, those underlying rules that cells use to build complex structures, to build us. Cracking that biological software, understanding how cells compute and build, has profound implications for developmental biology, regenerative medicine, maybe even understanding and treating cancer. It's about uncovering a whole new layer of biological information processing.
So, looking forward, what are the takeaways? If you're working in this field or just interested, what should the focus be? Seems like focusing on those contained applications, especially the biomedical ones using Anthrobots, makes the most sense. And crucially, developing those governance models now, alongside the science, not after the fact. Plus, managing public expectations, communicating accurately, being excited about the potential, but realistic about the timelines and hurdles. Balancing that hype and reality.
Exactly. And maybe the deepest takeaway is this. Xenobots are more than just a new gadget. They're really a new kind of question. They force us to ask. What else is life capable of that we haven't even imagined? How do biological systems compute, solve problems, organize themselves in ways we're just starting to glimpse? And maybe the biggest question, how can we learn to partner with that innate collective intelligence of cells to tackle our biggest challenges, from disease to, yes, maybe eventually environmental issues?
It really does make you wonder, doesn't it? Beyond frog cells, what are the biological systems? Maybe microbial communities, maybe plant tissues, maybe whole ecosystems hold untapped computational or organizational secrets we could potentially learn from or even partner with in the future. Lots to think about there.